Yeah, so the ultra-low-pass whole genome sequencing is a new technology that has been invented by the liquid biopsy core at the Broad Institute in Cambridge. And it's a very interesting tool because we can sequence at a very low pass. It's 0.1x. And what we get from that for the cell-free DNA actually is we can detect the copy number abnormalities. And from that, we can infer the tumor fraction within the cfDNA. So we sequenced 88 samples from myeloma patients, the cfDNA, right? And we found in 75 of them more than 3% of tumor DNA within the cfDNA. So it's a way to assess if there is enough tumor DNA in the cfDNA to further sequence the samples, basically. Multiple myeloma is characterized by clonal heterogeneity and clonal evolution. So the clonal evolution induces like progression of the disease and relapse after treatment and treatment resistance. So there is a real interest to be able to follow the patients longitudinally for their genomic features across the course of the disease, right? And so what we did here is we used the cfDNA liquid biopsy to assess the genomic abnormalities in myeloma patients. But for that, the first study that we did and we presented here was to compare cell-free DNA to the bone marrow tumor cell DNA, and we did a whole-exome sequencing for that. So we had 10 patient samples with matched cfDNA and the bone marrow tumor cells. We applied the whole-exome sequencing on them, and we compared them. So basically, in terms of copy number, alterations, and the main somatic mutations, it was pretty comparable, and we could identify in both compartments the mutations or the copy number alterations. What's interesting as well is that we looked at the clonal heterogeneity in these samples, and we inferred the clonal and subclonal mutations in both of them. We could identify 100% of the clonal mutations in both cfDNA and the bone marrow tumor cells, and it was about 2/3 of the subclonal mutations. So, which is interesting because it means that first it's a good proxy, cfDNA is a good proxy of the bone marrow tumor cells first, but it also can improve the clonal heterogeneity definition in multiple myeloma. We believe that these need to be further assessed in prospective clinical trials, right? And the goal would be to have sequential samples across the course of the disease and being able, in that way, instead of doing multiple bone marrow biopsy, for example, and being able to follow the clonal evolution of the patients and to potentially detect mutation resistance early on and be able to adapt the therapy and the treatments based on that. So it's really like the possibility to do multiple blood samples to sequence these samples and have a real vision of multiple myeloma at the genomic level. Absolutely. So instead of doing multiple biopsies, we just need a simple blood sample to do this sequencing, and we have all the mutations that are present in the bone marrow, and even potentially more, because the bone marrow biopsy is just a one-side biopsy, and in the blood we can potentially get different clones that are in a separate side. So it can even like improve the definition of the clonal heterogeneity. So the ultra-low-pass whole genome sequencing is very low-cost, actually, technology. It's only like $20 per sample, so which is very affordable. And then a targeted deep sequencing could be also low cost based on new technologies that we have, and can sequence very deep for affordable cost now. Looking forward to develop this in clinical trials. We are working on that with the French team actually now, so hopefully we'll be able to present new data about this very exciting technology in the near future.